food on your plate was edited in a lab before it ever touched soil. Not cooked, not seasoned, genetically engineered. We're talking about bioengineered food. Crops whose DNA has been altered by scientists to grow faster, resist pests, survive droughts, and even produce their own pesticides. Supporters say this technology could solve world hunger. Critics say it's turning our food system into a corporate science experiment. So today, we're breaking down both sides. First, how bioengineered food is actually made, from lab to table. Then, the part most people don't talk about, the dark side behind it. Part 1, how bioengineered food is made, from lab to table. Step 1, finding the desired trait. Scientists start by identifying what they want the plant to do better. This could be drought resistance, insect resistance, faster growth, or enhanced nutrition. They search nature for organisms that already have this trait, often bacteria. For example, a soil bacterium naturally toxic to insects have been used to engineer pest-resistant crops. Step 2, gene extraction. Once the target gene is identified, scientists isolate it using molecular enzymes that cut DNA at precise sequences. This extracted gene becomes the blueprint scientists want to insert into the plant genome. Step 3, gene insertion. There are two major methods for this. One, being the bacterial vector method, using agrobacterium to transfer DNA into plant cells. Or 2, the gene gun method, which is a microscopic DNA-coated particles that are shot into plant tissue. If successful, the plant cells integrate the new genetic code. Step 4, regeneration. Modified cells are grown in sterile lab environments using plant hormones. These cells regenerate into full plants carrying the engineered trait. Step 5, field testing. Plants undergo years of trials measuring the yield, stability, environmental interaction, pest resistance, cross-pollination risk. Step 6, regulatory approval. Agencies evaluate the allergenicity, toxicity, and nutritional equivalence before approval. This process can take several years. Step 7, farm to table. Once approved, seeds are sold commercially. Bioengineered crops are processed into oils, syrups, cereals, snack foods, and animal feed, making them widespread in modern diets. Part 2, the dark side of bioengineered food. Corporate control of seeds. Patented seeds prevent farmers from saving or replanting harvest seeds. They must repurchase annually and sign licensing agreements. Herbicide dependence. Many engineered crops are designed to survive herbicides. This has increased herbicide usage and contributed to herbicide-resistant superweeds. Environmental biodiversity risks. Cross-pollination can transfer engineered genes into wild plants, potentially disrupting ecosystems and reducing biodiversity. Pest-resistant evolution. Insects exposed to engineered toxins may evolve resistance, reducing effectiveness and increasing pesticide reliance again. Long-term health unknowns. While regulatory bodies deem current GM foods safe, critics cite limited long-term human consumption studies and potentially allergy or microbiome impacts. Economic pressure on small farmers. High seed costs require chemical pairings and licensing fees creates financial strain, especially in developing regions. Balancing the argument. Supporters emphasize benefits, which are higher yields, climate resistance, reduced crop loss, enhanced nutrition, and lower land use. The debate centers on ethics, regulation, and long-term sustainability, not just the technology itself. Bioengineered food is already embedded in our food system. The question isn't whether we keep engineering food, it's how far we go and what controls the science behind what we eat.